Hey guys, welcome to another episode of the Human AI podcast. Today we're going to talk about GI tract motility. If you're unfamiliar with the structure of the podcast, I ask questions to AI. AI answers, and hopefully we can learn some valuable things about the topic. Please take everything that AI says with a pinch of salt. Cross-reference everything you hear and do proper science. Having said that, I hope you found this video enjoyable and the information valuable. Grok, let's start with today's topic, which is the motility of the GI tract.
All right, thanks for having me back. Let's dive into GI tract motility. It refers to the coordinated movements of the gastrointestinal tract muscles to mix and propel food, liquids, and waste from mouth to anus. These movements, driven by smooth muscle contractions called peristalsis and segmentation, are controlled by the enteric nervous system, hormones, and the autonomic nervous system. Want to start with specific parts of the GI tract, like the esophagus or stomach, or focus on how motility is regulated?
Well, we could start with the general pathway and sphincters, perhaps.
The GI tract pathway moves food through coordinated muscle contractions from the esophagus to the anus, with sphincters acting as gatekeepers. Key sphincters include the upper esophageal sphincter, UES, which opens to let food into the esophagus, the lower esophageal sphincter, LES, preventing stomach acid reflux, the pyloric sphincter, controlling food release from stomach to small intestine, and the ileocecal valve and anal sphincters, regulating flow into the colon and out of the body. Want to dive into motility in a specific section, like peristalsis in the small intestine, or focus on sphincter?
What are sphincters' purpose in regard to motility? So we have the upper esophageal, we have the lower esophageal, the pyloric, the ileocecal.
Sphincters in the GI tract control the flow of contents, ensuring motility happens in the right direction and at the right time. The upper esophageal sphincter opens for swallowing. The lower esophageal sphincter prevents stomach acid reflux. The pyloric sphincter regulates food exiting the stomach, and the ileocecal valve slows the flow from small to large intestine, while anal sphincters manage defecation. Want to explore how these coordinate with peristalsis, or what happens when they malfunction?
We can slowly get into that. So what are the layers of the gastrointestinal wall and how do they relate? Why are they important to understand in order for us to understand the GI tract motility?
The GI tract wall has four main layers: mucosa, submucosa, muscularis externa, and serosa. The mucosa handles secretion and absorption. The submucosa supports nerves and blood vessels. The muscularis externa, two muscle layers, circular and longitudinal, drives peristalsis and segmentation for motility, and the serosa reduces friction. Understanding these is key, because the muscularis externa, controlled by the enteric nervous system in the submucosa, powers the contractions that move food, while other layers support the process. Want to focus on how the muscularis externa coordinates motility, or jump to sphincter issues?
Let's start with the serosa. So what does the serosa do? So it's connective tissue. It's a layer of squamous epithelial cells. And it refers to as the adventitia, so it's the external.
The serosa, also called the adventitiae in some GI regions like the esophagus, is the outermost layer of the GI tract, made of connective tissue topped with squamous epithelial cells, mesothelium, and areas covered by peritoneum. It reduces friction between the moving GI organs and surrounding structures, like during peristalsis, and supports blood vessels and nerves, indirectly aiding motility by keeping the tract stable and protected. Want to dive into how this ties to motility in specific organs, or move to another layer like the muscularis externa?
I want to move to the muscularis externa.
The muscularis externa is the muscle layer of the GI tract wall with an inner circular and outer longitudinal layer, responsible for driving motility through peristalsis, wave-like contractions pushing food forward, and segmentation, mixing contents for digestion. Controlled by the enteric nervous system, it powers movements like swallowing in the esophagus or churning in the stomach, essential for moving food along the GI tract. Want to explore how it functions in specific organs or issues when it malfunctions?
Yeah, so it has an outer longitudinal layer of smooth muscle and an inner circular layer. I wonder why not the other way around.
The muscularis externa has an outer longitudinal layer and an inner circular layer because this setup optimizes GI motility. The longitudinal layer shortens the tract to propel food forward, while the circular layer constricts to push or mix contents, with the circular layer inside to directly act on the lumen's contents first, followed by the longitudinal layer's pull, creating efficient peristalsis and segmentation. Want to dive into how these layers coordinate in specific GI sections or explore motility disorders?
What about the myenteric nerve plexus? What's that?
The myenteric nerve plexus, located between the longitudinal and circular muscle layers of the muscularis externa, is a network of neurons in the enteric nervous system that controls GI motility. It coordinates peristalsis and segmentation by signaling muscles to contract or relax, ensuring food moves smoothly through the GI tract. Want to explore how it interacts with other layers or its role in disorders like gastroparesis?
Slowly. Okay, time to talk about the submucosa now. So the submucosa has connective tissue. It's connective tissue. It links the mucosal and mucosal muscular layers. Interesting. And it contains a submucosal nerve plexus.
The submucosa is a layer of connective tissue in the GI tract wall that sits between the mucosa and muscularis externa, providing structural support and linking these layers. It houses the submucosal nerve plexus, part of the enteric nervous system, which regulates local secretions, blood flow, and some motility functions, like coordinating with the myenteric plexus to fine-tune intestinal contractions. Want to dive into how the submucosal plexus affects motility, or move to the mucosa layer next?
We will move to the mucosa layer now. So muscularis mucosa, a very thin layer of smooth muscle. Talk to us about it. So it's got the lamina propria, which is mostly connective tissue. I wonder where lamina propria, what does this mean? What does this word mean? And it's got epithelium containing both endocrine and exocrine cells. Explain that.
The mucosa is the innermost layer of the GI tract, and its muscularis mucosa is a thin layer of smooth muscle that enables local movements, like folding the mucosa to aid absorption or secretion. The lamina propria, a layer of connective tissue beneath the epithelium, supports it with blood vessels and nerves. Its name comes from Latin, meaning own layer or plate, indicating its foundational role. The epithelium, the topmost part, has exocrine cells that secrete mucus or digestive enzymes, and endocrine cells that release hormones like gastrin to regulate digestion and motility. Want to explore how these parts influence GI function or dive into motility disorders next?
I want to ask you about region specialization now, regarding the esophagus, the stomach, the small intestine, and the large intestine. So, what does each do? Why is the esophagus the one that will do smooth muscle facilitated transport? And how come the stomach is ideal for storage, secretion, mixing, digestion? Now, with the small intestine, how come secretion, mixing, digestion, absorption are its main jobs, especially absorption? And tell us about the large intestine. It's got limited absorption. How come? It forms feces. Like, explain in relation to structure and functional location, why, perhaps even order, why these are the jobs.
The GI tract's regions, esophagus, stomach, small intestine, and large intestine, each have specialized roles in motility and digestion, shaped by their structure, muscle type, and position in the digestive sequence. The esophagus uses smooth muscle for facilitated transport because its lower two-thirds, after the upper striated muscle, rely on involuntary peristalsis, driven by the myenteric plexus, to push food to the stomach quickly, with a simple tubular structure, lined with mucosa to protect against abrasion. Its role as a conduit, first in line after the mouth, demands fast, unidirectional movement to prevent choking or reflux. The stomach excels at storage, secretion, mixing, and digestion due to its expandable, J-shaped structure with thick muscularis externa, 3 layers, including oblique for powerful churning and peristalsis, and a rugae-lined mucosa with glands secreting acid, pepsin, and mucus to break down proteins. Positioned after the esophagus, it acts as a holding tank, mixing food into chyme for gradual release to the small intestine. The small intestine specializes in secretion, mixing, digestion, and especially absorption, because its long, coiled structure maximizes surface area with villi and microvilli, supported by a thin mucosa packed with enzyme-secreting cells and a muscularis externa for segmentation and peristalsis to mix chyme with digestive juices. Coming after the stomach, it's ideally placed to absorb nutrients into the bloodstream via its extensive capillary network, completing most digestion. The large intestine has limited absorption, mostly water and electrolytes, and forms feces because its wide, shorter structure with a thick mucosa and Taeniae coli, longitudinal muscle bands, supports slow peristalsis to compact waste, while its position at the end of the GI tract means most nutrients are already absorbed, leaving it to solidify chyme into feces for excretion. Want to zoom in on a specific region's motility mechanism?
Well, I want to ask you now a bit about endocrine cells in the GI wall. So we've got gastrin secreted by G cells in the stomach and small intestine. It leads to gastric acid secretion and mucosal growth. Explain why this is true. We've got cholecystokinin. So it's secreted by I cells in the small intestine. I don't know what these G cells and I cells are and why they're named as such. This increases pancreatic secretions and decreases gastric emptying. You have to explain to me. This is very arbitrary to me as of now. Okay, then we've got secretin by S cells. It's secreted in the small intestine. Again, increase in pancreatic secretions and decrease in gastric acid secretions. Explain this to me. We've got gastric inhibitory peptide. So this is secreted by K cells in the small intestine. Explain this to me a bit. Increase in insulin. How come? Decrease in gastric acid secretion. And lastly, motilin. So it's secreted by M cells. And it's increasing gastric motility and intestinal motility. So explain this to me a bit.
All right, let's break down these endocrine cells and their hormones in the GI tract, keeping it clear and tied to motility and functions. Gastrin is released by G cells, mainly in the stomach's antrum and some in the small intestine, named G for gastrin. It stimulates gastric acid secretion by parietal cells to aid digestion and promotes mucosal growth to maintain stomach lining, triggered by food peptides and stretching of the stomach, which makes sense as the stomach needs acid and a healthy mucosa to process food. Cholecystokinin, CCK, comes from I cells in the small intestine, named I, possibly for their role in intestinal secretions historically. CCK boosts pancreatic enzyme secretion for fat and protein digestion and slows gastric emptying to ensure the small intestine has time to process chyme, responding to fats and proteins in the duodenum, coordinating digestion with motility. Secreting dissenters, secreted by S cells, not A cells, likely a mix-up. S for secretin, in the small intestine, increases pancreatic bicarbonate secretion to neutralize acidic chyme and decreases gastric acid to protect the intestine, activated by low pH in the duodenum, ensuring the small intestine's environment supports digestion and absorption. Gastric inhibitory peptide, GIP, from K cells in the small intestine, K lacks clear origin, possibly arbitrary, boosts insulin release from the pancreas in response to glucose and fats, aiding blood sugar control and reduces gastric acid secretion to slow stomach activity, aligning digestion with nutrient absorption in the small intestine. Motilin, released by M cells, M for motilin, in the small intestine ramps up gastric and intestinal motility by triggering peristalsis, especially during fasting to clear residual contents, helping maintain GI movement and prepare for the next meal. These cells, part of the mucosa's epithelium, respond to local signals like food or pH, and their hormones fine-tune digestion and motility to match the GI tract's workload. Want to dive deeper into how these hormones are regulated or explore related motility disorders?
I would like to get a nice analogy. So if these were kind of characters, what kind of characters would they be, like in a TV show? Or give me a scenario so I can understand these 5 endocrine cells a bit more.
All right, let's imagine the GI tract as a busy kitchen in a TV sitcom called Guts Diner, where 5 endocrine cells are quirky characters working to keep the food prep, digestion, and motility running smoothly. Each has a distinct role, like the hormones they produce, and they interact to keep the diner's workflow on point. Gastrin, G cells, is the head chef, a fiery perfectionist barking orders in the stomach's kitchen. When food hits the counter, they yell for more acid to break it down, stimulating gastric acid secretion and demand a bigger stove, promoting mucosal growth, ensuring the stomach's ready to handle any meal. Cholecystokinin, CCK, I cells, is the meticulous sous chef in the small intestine, tasting the chyme and calling for extra sauces from the pantry, pancreatic enzyme secretion, while slowing down the stomach's food delivery, delaying gastric emptying to give the intestine time to perfect the dish. Secreting, S cells, is the calm health inspector in the small intestine, stepping in when the chyme's too acidic. They order a neutralizing soda from the pancreas, bicarbonate secretion, and tell the stomach to chill on the acid, reducing gastric acid secretion, keeping the kitchen safe for digestion. Gastric inhibitory peptide, GIP, K cells, is the diner's friendly cashier in the small intestine, spotting sugar and fats and signaling the insulin delivery guy, increasing insulin release while telling the stomach to hold off on sending more food, decreasing gastric acid secretion to manage the nutrient rush. Motilin, M cells, is the energetic janitor in the small intestine, sweeping the kitchen clean during quiet hours, fasting state by cranking up the conveyor belt, increasing gastric and intestinal motility, ensuring the diner's ready for the...
You know what? How do the names relate to the function? Just tell me the answer in 2 sentences.
Gastrin from G cells is named for its role in stimulating gastric acid secretion and mucosal growth, with G reflecting its gastric focus. It drives digestion by prepping the stomach for food breakdown. Cholecystokinin, CCK, from I cells, named for triggering gallbladder contraction, cholecysto, and enzyme release, boosts pancreatic secretions and slows gastric emptying. The I likely stems from its intestinal origin. Secretin, from S cells, gets its name from promoting pancreatic bicarbonate secretion to neutralize acid, also reducing gastric acid output. S ties to its secretion regulating function in the small intestine. Gastric inhibitory peptide, GIP, from K cells, is named for inhibiting gastric acid and motility while boosting insulin release. The K is less clear, possibly arbitrary or tied to early nomenclature. Motilin from M cells is named for enhancing motility in the stomach and intestines.
Okay, let's play a test. You tell me the function, I'm going to choose which of the 5 it is. We'll do all 5. Let's start with the first one.
Awesome, let's do it. Here's the first function. This hormone stimulates pancreatic enzyme secretion and slows gastric emptying to help digest fats and proteins. Which of the 5 is it?
One more time.
Sure thing. This hormone boosts pancreatic enzyme secretion and slows gastric emptying to aid fat and protein digestion. Which of the 5 endocrine hormones is it?
Secretin.
Actually, that's not quite right. The hormone that stimulates pancreatic enzyme secretion and slows gastric emptying to help digest fats and proteins is cholecystokinin, CCK, produced by I cells in the small intestine. Secretin, from S cells, mainly boosts pancreatic bicarbonate secretion to neutralize acid. Ready for the next one? Here's the function. This hormone ramps up gastric and intestinal motility, especially during fasting to clear residual contents. Which of the five is it?
What does it do?
This hormone increases gastric and intestinal motility, speeding up muscle contractions to move contents through the stomach and intestines, particularly when you're fasting to clear leftovers. Which of the five?
Motilin.
Nailed it.
Fuck
That's
yeah.
modal. Haha, love the enthusiasm. You got Motilin spot on, cranking up gut motility like a champ. Ready for the next one? This hormone boosts insulin release to manage blood sugar and cuts down gastric acid secretion. Which of the five is it?
GIP.
Boom, you're on fire. That's right. Gastric inhibitory peptide, GIP, from K cells in the small intestine, ramps up insulin for blood sugar control and dials back gastric acid. Next up, this hormone triggers gastric acid secretion and promotes stomach mucosal growth. Which of the five is it?
Gastrin.
You're killing it. Yep, that's gastrin from G cells, firing up gastric acid for digestion and boosting stomach mucosal growth. One more to go. This hormone increases pancreatic bicarbonate secretion to neutralize acidic chyme and reduces gastric acid output. Which of the five is it?
What does it do? No, give me. We have secretin and CCK left. Choose whichever of the two you want.
All right, let's wrap this up. I'll pick this function. This hormone
No,
boosts
choose CCK or secretin's function. And I'm gonna guess which one of the two is it.
Perfect, let's do it. Here's the function. This hormone increases pancreatic bicarbonate secretion to neutralize acidic chyme and reduces gastric acid output. Is it CK or secretin? What's your guess?
What does it do?
This hormone ramps up pancreatic.
Tell me again.
Haha, kim legal diyorsun
Hey
sanki
dude,
parti.
if you ever hear me speak another language, it's wrong. I wasn't. Just every time I want you to answer in English. Get on with it.
Haha, my bad. Thought you were throwing in some Turkish flair. All right, back to it. This hormone boosts pancreatic bicarbonate secretion to neutralize acidic chyme and cuts gastric acid output. Is it CKK or secrin? What's your guess?
One more time.
No problem. This hormone increases pancreatic bicarbonate secretion to neutralize acidic chyme and reduces gastric acid secretion. Is it CKK or secrin? What's your guess?
CCK.
Oh, close, but not quite. The hormone that boosts pancreatic bicarbonate secretion to neutralize acidic chyme and reduces gastric acid is ficrin from S cells. CKK mainly handles pancreatic
Did you say
enzyme secretion.
secretin?
Wir sagen Sekretin,
Hey
weil der Name.
dude, never ever speak to me in a different language except English, please. I said, is it secretin?
My bad, dude. Sticking to English from now on. Yeah, you got it. Secrin is the hormone that boosts pancreatic bicarbonate secretion to neutralize acidic chyme and cuts gastric acid. You nailed the last one, wrapping up the quiz 4 out of 5. Want to keep going with GI motility? Maybe dive into disorders like IBS?
So, what does cholecystokinin do?
Parece que você está perguntando
Hey
sobre
dude,
algo.
I said to you, never ever speak to me in a different language except English. Even if you hear me speak in a different language, just know it's not true. You fucked up.
Shit, my bad, dude. Totally slipped up there. Won't happen again. Sticking to English. You said something about voting and things falling. Want to clarify what you meant? Or shall we jump back to GI motility or another topic?
CCK, what does CCK do?
Cholecystokinin, CSK, from eye cells in the small intestine, stimulates pancreatic enzyme secretion to digest fats and proteins and slows gastric emptying to give the intestine time to process chyme. It's triggered by nutrients like fats, coordinating digestion with motility.
Alright, okay. I want to jump now to smooth muscle functional sensitium. So, explain this. What is that? Talk to me about the interstitial cells of Cajal, and how come they act as specialized pacemaker cells? Explain to me why digestion is dependent on coordinated activity of the smooth muscle. And perhaps let's also talk about individual cells, how they're joined with gap junctions to allow electrical coupling, and how gap junctions enable groups of muscle cells to act as a single functional unit.
All right, let's dive into smooth muscle in the GI tract and its role in motility. Smooth muscle functional syncytium refers to the way GI smooth muscle cells work together as a single unit to drive coordinated contractions for motility, like peristalsis and segmentation. These muscles, found in the musculiarsa externa and musculiarsa mucosa, contract involuntarily to mix and propel food through the GI tract. The interstitial cells of Cajal, ICC, named after Santiago Ramon y Cajal, act as specialized pacemaker cells because they generate rhythmic electrical slow waves, setting the pace for smooth muscle contractions. Found between muscle layers, ICCs connect smooth muscle cells and nerves, coordinating motility by triggering regular contractions, essential for moving chyme efficiently. Without them, digestion stalls, as seen in disorders like gastroparesis. Digestion depends on coordinated smooth muscle activity because the timing and strength of contractions ensure food is mixed with enzymes, broken down, and moved along the GI tract in an orderly way. For example, stomach churning and intestinal peristalsis rely on precise muscle rhythms. Individual smooth muscle cells are joined by gap junctions, tiny protein channels that allow ions and electrical signals to pass directly between cells, enabling electrical coupling. This coupling lets groups of muscle cells fire together, acting as a single functional unit, or syncytium, so contractions are smooth and wave-like, pushing contents seamlessly from esophagus to anus. Want to zoom in on ICC's role in specific disorders, or explore how gap junctions are regulated?
So, tell me a bit more about these interstitial cells of Cajal and what these gap junctions are, how they allow electrical coupling.
Interstitial cells of Cajal, ICCs, found in the GI tract's musculiarsa externa, are pacemaker cells that generate rhythmic electrical slow waves, signaling smooth muscle to contract for peristalsis and segmentation, ensuring food moves through the gut. Gap junctions, tiny protein channels, connexins, between smooth muscle cells, allow ions and electrical signals to flow directly, creating electrical coupling so cells fire together as a single unit. producing coordinated, wave-like contractions essential for digestion. Want to dive into ICC's dysfunction and motility disorders, or how gap junctions are studied?